More than 1 in 7 Americans suffer from chronic kidney disease, according to the CDC, with the best treatments for advanced stages including dialysis and transplant, forcing patients to rely on the generosity of their friends and family while in constant fear of organ rejection. My name is Sophia Lehman, and as a Huang Fellow in the Musa Lab, I am working to give patients treatment options which will allow them to live their life free from this fear. Welcome to a conversation with my lab mentor, Rohan Bhattacharya, who has already made significant steps toward this goal.
Hi Sophia, I am Rohan Bhattacharya. I'm a second-year PhD student working in the laboratory of Dr. Samira Musa in the Department of Biomedical Engineering at Duke University.
Could you tell listeners what we do at the Musa Lab?
Our lab is focused at the interface of engineering and medicine. We harness different concepts of biology, including developmental biology, stem cell biology, and microfluidics to model stem cell development. And our lab recently discovered a method for the derivation of human podocytes, which are one of the most important cell types of the human kidney glomerulus. When we talk about glomerulus, you can think about a tight molecular sieve. And this sieve is responsible for size-selective filtration of toxins from blood. It is also important to know that these cells are actually terminally differentiated. That means they don't have the capacity to regenerate. So if you are undergoing any treatment, like maybe chemotherapy, or if you have some mutations in your DNA, these cells simply detach from the blood capillaries of the glomerulus. And this unfortunately exposes a bigger part of the blood capillary to the urinary compartment, which ultimately leads to chronic kidney disease, which is an epidemic in the United States. That's why there is a bigger need to understand how these organs develop and what kind of molecular mechanisms are associated with disease progression, disease development that could allow us to model these diseases outside human body with the goal of therapeutic discovery.
In our lab, this model is called an organ on a chip, which integrates human-induced pluripotent stem cells and microfluidic engineering to imitate the function of human organs.
Our organ on chip will allow you to model diseases and discover certain treatment regimens that are very specific to a particular patient or a particular population, but not be reflected in animal model studies.
Could you describe why animal models are a poor proxy for human pathology?
If I talk about the kidney, the developmental timeline for kidney is quite different between human and mice. So that was the motivation behind developing new differentiation strategies from human iPS cells that could help us model organ development and also model disease mechanisms within these tissue chips.
You speak with such passion about kidney pathology. Do you know anyone who has suffered from chronic kidney disease?
Actually, one of my cousins passed away from chronic kidney disease, and it's probably one of the most ignored disease because 90 to 95% of the patients do not know they have the disease until and unless a substantial damage to their kidneys has occurred. And this is primarily because we don't have a biomarker yet that can help in early diagnosis of this disease. And a discovery of a biomarker to detect chronic kidney disease is only possible when you're aware of the molecular mechanisms involved in the disease development. And I believe we would be able to do that only when you couple multiple mature cell types together by using an organ-on-chip platform.
This was Combating Kidney Disease with Rohan and Sophia.